Today we will discuss intrapopulational variations and comparison of population samples. Basically, the traditional method of classical taxonomy was to group individuals which are found at a given locality uh, by looking at the similarity among those particular organisms and call each similarity class a different species. So basically, one of the primary tasks of the taxonomist is to unmask such variants and assign them to species to which they belong. We have a number of variants in a population, and those variants may be on the basis of the sexual differences, or those may be on the basis of the age differences, and there are many other causes of the variations. So due to this, it becomes the primary task of the taxonomist to unmask these variants, and we need to assign them species to which they belong. And why we want to assign species to these variants? Because we want to get maximum benefit out of all these individuals, not only in terms of our on the applied side, but also on the basic side. We need to have all these species which have been created by the nature and created by the prime authority, or created by Allah, that has been a need of our survival. Because if all the species will survive, the ecosystem um, will be balanced, and due to this we need to identify them, and we need to understand their status, and we need to conserve them. So there are different reasons why we study taxonomy, and during working on different samples, we face different intrapopulational variants, and we need to compare these population samples to reach to a conclusion that to which particular species an animal belongs to. So different fauna within sympatric population samples are the source of confusion. There are three reasons of the confusion. Number one, an extreme difference in fauna belonging to a single species. Number two, an extreme similarity of good biological species, that is sibling species. And number three, an extreme variability and wide phenotypic overlap of the two species. Basically, there are two types of intrapopulational variations which are carefully evaluated by an experienced taxonomist. These are genetic variations, and these are non-genetic variations. For the identification purpose, we use two criteria. Number one, morphology, and number two, reproductive isolation. The members of a population may be morphologically identical, or they may be different. So if we are considering morphologically identical individuals, they might be having reproductive isolation, or they may not be having reproductive isolation. If they are having reproductive isolation, then they are called sibling species. The members of a population that are having identical morphology, however they are reproductively isolated, it means they belong to sibling species. And if they are not reproductively isolated, they are called same fanon of a single species. On the other hand, if the morphology of the individuals of a population is different, then they are not reproductively isolated, it means that they are different fauna of the same species. And if the individuals which have different morphology are reproductively isolated, it means that they are different species. So the problem for a taxonomist, or the confusion for a taxonomist, occurs when there is the presence of sibling species, or there is the presence of different fauna of the same species. Because sibling species cause confusion as they are very identical, however they belong to different species. due to their reproductive isolation. On the other hand, um, if you are considering number 2, which is different fauna of the same species, that is also difficult to identify, because within the same species there are drastic differences in the fauna of the individuals, which is really confusing for the taxonomists to to reach to a conclusion. Various forms of individual variations are listed here. So, as we have previously discussed, that there are two most important types of variations. They are non-genetic variations and genetic variations. If we consider non-genetic variations, number one is individual variation in time. Individual variation in time has been further subdivided into three categories. That may be age variation, which is due to which is shown at the different phases of the life cycle. Then there are seasonal variation in an individual, and number 3 is the seasonal variation in consecutive generations. B is social variation. Social variation is represented by all the insects that are having caste system in their colonies. That may be ants, bees, and other animals like termites. C is ecological variation. Ecological variation may be due to the habitat variation, that is called ecophenotype. And then there is variation induced by temporary climatic conditions. Then there is host determined variation. Number 4, density dependent variation. Number 5, allometric variation. Number 6, neurogenic or neurohumoral variation. These variations will be discussed in detail in the further slides. Then there is traumatic variation, that is due to parasitic induction, and there is the parasite induced variation, or that may be accidental or teratological variation.
Individual variation in time. Number 1, it's age variation. So animals in general pass through a series of juvenile or larval stages in which they may be quite different from their adult forms, due to what there might be a confusion for the taxonomists while doing identification, and that may lead to the cases of synonymies. Synonymies, again, I tell you that it is naming of the same species multiple times. Multiple names of the same species is called synonymy. So therefore, the catalogues of any group of animal which have been developed in the past may list numerous synonyms that result due to the failure of the taxonomists to recognize the relationship between various age classes of the same species. In reptiles, birds, mammals, there are no larval stages, but immature individuals may be very different from those of adults, particularly in the case of the birds. Several hundred bird synonyms which have been based on the juvenile plumage have been found in the catalogues, and in many fishes there are immature forms which are too much different, which are drastically different from those of their adult forms, and due to what they have been placed in different genera and even different families. Similarly, there are certain problems of neotenic animals. Neotenic animals are those animals that are sexually mature in the larval stages, and due to what that causes confusion, and the taxonomists put them into different species. So the difficulties for the taxonomists are even greater in the groups with larval stages, so which are quite different, having the faintest resemblance with the adult. For example, if you take the example of butterfly or frogs, tadpole is extremely different from the adult form. Caterpillars are extremely different from the butterfly. On the other hand, if you think of coelentrates, echinoderms, mollusks, crustaceans, their adult forms are extremely different from their larvae. So the taxonomic status of such larval stages can be settled either by assembling a complete sequence of intermediate stages or by rearing that particular organism through a complete series of the life cycle. And it is of extreme importance that we have to visit the animal, we have to see the animal, we have to observe that particular animal in their field, and for a longer period, for at least in which they pass through their reproductive phases, and which we need to observe them by looking at their life cycles or by rearing them in our labs. So the taxonomic identification of the larval stages of the parasites is also of great importance and is of great difficulty, in which different stages occur on different hosts. Age variation is not restricted to differences between larval stages and adults, but also it occurs between the youngs and old adults. For example, if you would take the example of deers, the older stags often have antlers with more points than those of the younger stags. The shape of the antlers may also change. This age variation must be considered when the authors of different species or subspecies are working on the antlers. So there is probably no further addition of points after a certain age has been reached. So it becomes futile when we try to determine the exact age of the stag by the number of points of its antlers. So similarly, if we are considering the rattlesnake, it becomes of no sense to determine their age using the number of rings in their rattle. In the cases of snakes and fishes, in which we generally observe a continuous growth, we need to find out certain characters which are of importance for the taxonomic characterization. And these characters are mostly meristic characters. For example, the number of scales or the number of fin rays that do not increase after they are formed in spite of the enormous subsequent growth. So these characters are especially useful in the herpetology and ichthyology. Seasonal variation in an individual. There are certain animals that live as adults through several breeding seasons. The same individuals sometimes have very different appearance in different parts of the year. During their breeding season, they have striking colors. On the other hand, when they are passing through a non-breeding phase, their plumage undergoes molting, and they are having much dull shades of their plumage. So similarly, if we consider the polar bears, they are, or not only the polar bears, but also Arctic and subarctic birds, there may be a change from a cryptic white winter dress to a normally colored summer dress, and the winter dress is extremely white and the summer dress is having a bit of yellowish tinge. So you can also take the example of the European starling. The freshly molted bird of the October, is covered with the white spots, and all the feathers show whitish or buffy margins. However, when there approaches the winter, the pale edges of the feathers wear off, and again in the spring, which is the beginning of the breeding season, the whole bird is beautifully glossy black. So a similar process of wearing begins in the males of the birds also, and it brings out full colors of the nuptial plumage in the males. In arid regions, particularly in real deserts, the sun bleaches the pigments, A bird before the molt looks much paler than does in freshly molted plumage. Such seasonal variations are of particular importance and are common among the vertebrates because of their elaborate endocrinal system. So, due to all these factors, many seasonal variants are described as distinct species, which is not true, definitely, because they are the members of the same species in different seasons. So it is extremely important that we have to, again, I'm saying the same thing, that we have to observe the animal in their fields, where in their particular habitat, throughout their breeding and non-breeding season, so that, or we can culture them in our labs so that we can look at them, look at their all life stages along with their all phases of life, which is the only solution to the problem of identification.
Seasonal variations in consecutive generations. Many species of short-lived invertebrates, particularly insects, produce several generations in the course of a year. In such species, it is common for the individuals that hatch in the cool spring to be quite different from those produced in the summers, or for the dry season individuals to be different from the wet season populations. Some tropical butterflies may have very different dry season and wet season fauna, like their appearance is quite different, or their phenotypic appearance, their phenotype is very different from between the dry season and wet season animals. Such seasonal forms can usually be recognized not only by the occurrence of the intermediates in the intervening season, but also through the identity of wing venation and genitalia. Then we have a concept of cyclomorphosis. It is a special kind of seasonal variation which is found in certain freshwater organisms, particularly rotifers and cladocerans. The population of a species undergoes regular morphological changes through the seasons in connection with the changes in temperature, turbulence, and other characteristics of the body. Many species have been named, particularly in genus Daphnia, that were nothing but just seasonal variants. In rotifers, different morphs have also been the result of different kinds of food they were consuming. Social variation, that is insect castes. In the social insects, such as some bees and wasps, but particularly among ants and termites, castes have been developed. There are definite groups of individuals within a colony. In addition to the reproductive castes, there are workers and soldiers, also sometimes of different types. In the Hymenoptera, these castes are most commonly modified females and are genetically identical. But in the Isoptera, to which termites belong, both sexes may be involved. The structural types observed may result from different larval food, or that may be due to hormonal or other controls. Obviously, taxonomic names should not be applied to these intracolonial variants, but invalid species have sometimes been described because it was not realized that there were different types of soldiers or workers in the same colony. So we will discuss ecological variation. First of all, we will discuss the impact of habitat variation on members of the same species in different habitats. Different populations of a single species that occur in different habitats of the same region are phenotypically different. What should be the taxonomic treatment of such local variants? Some authors describe them as different species, while others diagnose them as non-genetic variants. They may be micro subspecies, or they may be called as ecological races. And secondly, they may be the non-genetic ecophenotypes. A major example of such plasticity in phenotype is found in freshwater snails and mussels. The parts of the rivers which have cooler temperatures and flow rapidly have different forms than those which are at higher temperatures and with slower flow. Similarly, the areas with a higher concentration of the lime are different as compared to those found in the areas with lower concentration of the lime. Whether a given habitat form is an ecophenotype or a microgeographic race is not always evident, so it becomes necessary to transplant an organism or raise that particular organism in the laboratory to answer these questions, so that the confusion regarding the identification of a particular organism can be removed. Ecological variations induced by temporary climatic conditions. Some animals showing high phenotypic plasticity produce visibly different forms due to varied climatic conditions, that is drought, cold, or food availability, produced through the same year. For example, fish from a year class may be stunted or that may show a rapid growth depending on the conditions. A correct sampling of a particular species or of a particular fish, that is what that means, that is well sampled throughout the whole year, compensates for the confusions caused due to these distortions. You may have a question that what is a year class? It is a group of specimens produced in a particular part of a year. Host-determined variation. Host-determined variation in the parasites of plants and animals provides a potential source of taxonomic error and causes confusion with microgeographic races or sympatric species. A microgeographic race is a highly localized and distinguishably differentiated population within a natural species. However, sympatric species is generally defined as a new species from surviving ancestral species and inhabits the same geographical area. The phenomenon most commonly results in size differences and may also involve other morphologically and physiologically different characters. Host-induced variation is not as common as was formerly believed. Many so-called host races in the literature have been found to be valid sibling species. As we know that if we consider human beings, Homo sapiens, we have a number of races within the same species, and those races are not reproductively isolated from other races. So the microgeographic race is a highly localized and distinguishably differentiated population within a natural species. Density-dependent variation. The effects of crowding are sometimes reflected in morphological variation, especially when crowding leads to shortage of food materials. However, density-dependent variation need not to be related to the food supply only. For example, the gregarious species of locusts exist in various unstable biological phases. These phases differ in anatomy, color, and behavioral characteristics and have often been described as distinct species. When newly hatched nymphs are reared under crowded conditions, they develop into transitional phases. And when they were isolated and reared separately, they develop into solitary phases. Similar phases have been reported for two species of the army worms. Allometric variation. Allometric growth may result in disproportionate size of a structure in relation to the size of the rest of the body. If individuals in a population show allometric growth, animals of different size will show allometric variability. This phenomenon is particularly marked in insects, or it is prominent in insects. It involves such features as the head of the ants, the mandibles of the beetles, the frontal horns or thoraxes of the scarabs, and the antennal segments of the thrips. Failure to recognize the nature of such variations has resulted in synonymy. Neurogenic or neurohumoral variation. Neurogenic or neurohumoral variation refers to the color change in individual animals in response to the environment. These changes are accomplished through the concentration or dispersal of the color-bearing bodies called chromatophores. This type of variation was first thoroughly studied in chameleon. It occurs sporadically in the lower animals but is best developed among crustaceans, cephalopods, and cold-blooded vertebrates, that is cyclostomes, elasmobranchs, teleost fishes, amphibians, and reptiles. Traumatic variations vary in different groups. The abnormal nature of this type of variation is usually obvious. However, this might be cause of confusion while doing the taxonomic work. Apart from such familiar effects of parasitism such as swelling, distortion, mechanical injury, parasites may produce conspicuous structural modifications as well. In the bees, parasitism by stylopes frequently results in a reduction in the size of a head, enlargement of the abdomen, and changes in the pubescence and wing venation. It also commonly results in the formation of the intersexes. In females, it is found that it causes this parasite-induced induction causes reduction of the pollen collecting organs, loss of the anal fimbriae, changes in the relative length of the antennal segments, reduction of the facial foveae, reduction of the sting and accessory organs, paling of the ventral abdominal pubescence, acquisition of the angular cheeks, and yellow on the normally dark clypeus. However, in the males, it is reported that there is the development of long hairs resembling those of females, broadening of the posterior basidorsus, changes in the proportion of the antennal segments, loss of the cheek angles, and some yellow from the clypeus, indications of the facial fovea, and reduction in the size of the genitalia. Some striking different termite soldiers have also been reported, which were assigned to a new genus and species due to individual parasitism. Then there are post-mortem changes, which are also a cause of confusion for a taxonomist while doing identification. In many groups of animals, it is impossible to prevent post-mortem changes of preserved specimens. The deep orange-yellow plumes of the 12-wire bird of paradise fade to white in collections. The plumage of the Chinese jay, which is green in life, turns blue in collections because of the loss of the volatile yellow component, which is a part of the pigment. Many birds that are clear gray and or they are olive gray, when freshly collected, becomes more and more black due to the oxidation. So numerous synonyms have been created in ornithology due to the comparison of freshly collected material with that of the old museum specimens. Other post-mortem changes result from the chemical action of the preservatives or the killing agents. The common color change also takes place in certain insects. The specimens turn bright red and due to the reaction of the preservatives. So it is generally recommended that we should take photos, and preferably we should take colored photographs or make color sketches, so that these recordings will allow an accurate description of the living material and will be used for the comparison when required. Thank you so much, and if you do have any questions, you may ask it on the same forum, which will